This talk is about some of those more exotic, not so well-known viruses, which are the orthobunyaviruses. So, for those who study viruses, as you might know, in recent decades, we've had several issues which has contributed to the emergence of different arboviral diseases. We have had global warming and globalization. So we have people traveling back and forth on vehicles, on planes that carry vectors. We've had increases in temperatures that have also allowed vectors to spread and multiply in areas they might not have normally been seen, right? Because of the increase in human population, we've also had a lot of deforestation, which again has created areas that vectors and humans intersect even more than in previous decades. And the last half century, or the last half, I should say, of the 20th century, we saw an increase and emergence of different diseases, not just arboviral, but just viral diseases because of all these factors. So as we've had the experience of uncontrolled vector populations, we've seen the increase in the spread and the emergence of these viruses. So, as I said, every year, people have been discovering new viruses because of humans living on the intersect between what was normally very rural, untouched virgin forest that has now been invaded by our expansion as humans and as a society. So the question continues to be, what else is out there? So we're seeing all of these viruses coming out, things like Ebola, even as far back as HIV and the AIDS epidemic, were all emergent diseases caused by humans interacting with wildlife and their environment. And the question remains, what else is out there and how can that impact us as a society? So just to give you some background on this study, in the 1950s, the Rockefeller Foundation in the US set up several virus research labs globally in places like South Africa, India, Egypt, Nigeria, Brazil, Colombia, and in Trinidad. In Trinidad, they set up an institute called the Trinidad Regional Virus Lab, which was established in 1953. And several scholars who were well known in arbovirus studies came and were in charge of that lab. So we had people like Downs, Aikins, and Spence, who over the next 2 decades did a lot of large-scale surveys of wildlife and vectors found in forested areas in Trinidad, isolating some known arboviruses like yellow fever and equine encephalitis viruses, and also discovering new viruses that were novel at the time. So just to give you an idea, for those who don't know, this is Trinidad, and the Rockefeller Foundation lab mainly studied in this area of Trinidad. So when we zoom in on the map, what we have is a swamp area called Nariva Swamp, and beside it is what's known as bush forest, which is a protected reserve. So in that reserve, there is a lot of wildlife. So there are capuchins, howler monkeys, other animals like porcupines and opossums. So all of these animals were there, and they basically served as hosts for a lot of the viruses that were found. For example, the howler monkey has been known to be a host for yellow fever virus, and the same with the capuchins. In addition to these native species, Trinidad has also had other wildlife introduced from the mainland. So in the upper right, what we're seeing is what's called a tufted capuchin, which is an introduced species, but over the last few decades has expanded and established itself. So Trinidad is a country that has, despite it being in the Caribbean, it has a lot of vegetation that is characteristic of South America, and it has similar wildlife to the continent. And for that reason, there are a lot of potential host species here that could harbor these arboviruses over time. Now, the work that they did at the Trinidad lab, as I said, included finding several novel viruses, and key among these were orthobunyaviruses. So both in Trinidad and the lab in Brazil, they were finding these new species, some of them common to both countries. Now, at the time in the 50s and 60s, they characterized these viruses largely by serology, right? So when they described orthobunyaviruses, they had serogroups. And for many decades, until we had the improvements in sequencing technology, a lot of these viruses remain uncharacterized in terms of their genetics. I'm just showing you the pictures on the right are just showing you two of the scientists. On the top is Downs, the second person is Spence, and this is Mayarovirus, which was first isolated in Trinidad in the 1950s. So just to give you a background about orthobunyaviruses, for those who don't know, the orthobunyavirus is one of 8 genera in the Peribunyaviridae virus family. And that's according to the recent ICTV classification, right? They are widely distributed and they are almost exclusively arthropod-borne. The main vectors for these viruses are mosquitoes and ticks, and they're found in both hemispheres. And if you go on the ICTV website, you'll see that there are at least 138 viral species, which classifies over 200 different viruses. So there are a lot of viruses and they have a lot of genetic diversity, right? There are at least 18 serogroups that have been characterized, so they have the same serology, and traditionally, this is how the orthobunyaviruses have been grouped. So out of those 138 species, at least 30 have been shown to be pathogenic to humans. And these are just a few examples. There's lacrosse virus, which causes pediatric encephalitis, Oropouche virus, which was found in a place called Oropouche in Trinidad, which causes febrile illness. And there's Nagari virus, which was found, I think, somewhere on the African continent. I can't remember exactly where, which causes hemorrhagic fever. So, I mean, 30 species, that's a significant number, and they do cause illness in humans. Many of those 30 species fall into 5 of those 18 serogroups, and these are the main ones. We have what's called a Group C serogroup, Simbu, Bonyamora, California, and Wamba serogroups. There are other members that affect domestic species, for example, Ano and Cache Valley viruses, but our talk will mainly focus on the ones that we've seen that are in these pathogenic serogroups. So these orthobunyaviruses are envelope negative sense single-strand RNA viruses, right? They have a tripartite genome where they have a large segment RNA, a medium segment, and a small segment encoding different proteins, right? Large segment encodes the RNA-dependent RNA polymerase. Medium segment does glycoproteins for the viral surface and the non-structural protein NSM, while a small segment encodes the nuclear capsid, and in some species, it also encodes a smaller non-structural protein called NSS. And this is just a nice figure from viral zones showing what the genome looks like, right? So as we said, large, medium, and small segmented single-strand RNA genome. And once it infects, those go outside, they're encoded, replicated. The negative becomes positive, of course, which is how those RNA viruses replicate. And then you can get transcription of the different viral proteins. So because these viruses have this tripartite genome, in addition to the normal mutation rates that you get with RNA viruses, which are usually relatively high, we also get genome reassortment naturally, right? So like influenza, because you have separate segments, if you have multiple species in the same host, these viruses can basically trade segments, right? And that has always been an issue when it came to doing the phylogenetics of these viruses. Because in addition to just looking at their microevolution, you also have to account for their ability to have reassortment. And this is a good figure from a paper by Nunes et al. In 2005, showing the potential reassortment. So if you have two different parental viruses, 1 and 2, in the same host, you can end up with 8 different viruses in terms of their genome. You can have the original parent 1 and parent 2, and then 6 other variations due to reassortment. So that means these viruses can have a lot of genetic diversity depending on whether they co-infect hosts or not. So traditionally, because of this high genetic diversity, these viruses have always given trouble to sequence fully, right? For that reason, in the genetic database, historically, you would have a paucity of reference sequences for the genera. But as we know, the last 10 to 15 years, we've had a great advance in next-gen sequencing, and that has allowed people to go back to these reference viruses, these isolates, and try to resequence them to try and more fully characterize these orthobunyaviruses. And there have been several efforts over the past 15 years where people have gone back to reference genomes, reference isolates, and tried to better characterize them to map their evolution to a greater degree. So our group had a few research questions that we wanted to answer. The first was, which orthobunyaviruses were still in circulation in Trinidad? The fact of the matter is, after the first 2 decades of work by the Rockefeller TRVL lab in the 50s and 60s, not a lot of work was done in the ensuing decades. So that was one question. The second was, do the genetic characteristics we can see, does it match or how does it relate to those orthobunyaviruses first sequenced back in the 50s and 60s, right? So if we found anything currently, how did they relate to those first reference isolates? Another question we had was, does the genetics actually match the serology? Now, one thing you'll see, particularly in viruses that are closely related, is a lot of cross-reactivity. And we see that in some flaviviruses like dengue and yellow fever and so on. And the question was, these viruses were originally characterized by serology and separated that way. Do those serocomplexes still hold now that we have greater sequencing technology that can better characterize these viruses? So that was another question we had. The last question we had were any of the species or isolates that we would be able to look at natural reassortants? So with those questions in mind, we had these hypotheses. The first was that the orthobunyaviruses discovered back in the 50s and 60s are the progenitors of any that we would find currently circulated in Trinidad. And we expected that we would find natural reassortants among whatever isolates we were able to identify. Right? A lot of this work was done through as a good colleague of mine called Alberto Guis, who did his PhD in the lab of Christine Carrington from 2007 to 2011. So I was working with them at times, and what we do is they would go into 2 different reserves, the Bush forest, which we saw earlier, which is where they did the work in the 50s and 60s, and another protected reserve called Aripo Savannah, where we would capture and screen those mosquitoes. Once we had pools of those vectors, we tried to do viral isolation sequencing and then characterization. So just to revisit this map, so this was the bush forest and Nariva swamp. So that was one study site, the same site that was used in the 50s and 60s. And then a second site, which was further north, another forested area called Aripo Savanna. So these were two protected reserves. So they had basically preserved the wildlife in these two areas. So the chance of finding the virus here was high simply because of them being protected reserves. So what we did, we got isolates from the World Reference Center for Emerging Viruses and Arboviruses at UTMB, Texas. And then using the pools that we surveyed, we extracted everything, got the nucleic acid, and then performed next-gen sequencing on those isolates to try and characterize the tripartite genome. Once we had the sequences, we did our phylogenetic analysis. Now, the first thing we did was use amino acids simply because of the high genetic diversity of these viruses. Once we were able to confirm phylogeny using amino acids, we then did the same with nucleotides, right? We also did some calculations of evolutionary rates using software such as BEAST, and we used the nucleotide data sets for that. So this table just shows the sequences that we got. So we managed to sequence 11 isolates, all right? 8 of them were from Trinidad, 3 from elsewhere, French Guiana, Panama, and Guatemala. And at least 8 of them, sorry, not 8, sorry, at least 4 of them were contemporary strains. And this is the first phylogeny I want to show. So this is of the L segment. So because of the tripartite genome, you have to do phylogeny of each segment of the virus to get a proper understanding of the microevolution. So looking at the L segment, what we saw is that after doing bootstrapping neighbor joining trees, our 11 sequence isolates fell into 6 serogroups, or 6 branches that were related or equivalent to the serogroups that were already characterized. Most of our viruses fell into what's called the group C serogroup. So we had 6 isolates in there, and then we had 1 isolate in 5 other groups: Patois, Simbu, Guama, California, and Bunyama serogroups. So a lot of the viruses we looked at were group C viruses. And as I said before, the group C is associated with pathogenic in humans, so that was pretty important. When we looked at the M segment, we saw a similar topology. So the same 6 isolates we saw before that were in group C fell into group C again, and the other 5 isolates matched the same serogroups as before. And we saw the same in the S segment, right? So 6 in group C and 5 distributed among 5 other serogroups. So in terms of that result, what we saw was that the phylogeny of these isolates broadly match those historical serogroups. So the serogrouping held for the viruses on a general level, and it was consistent across all 3 segments of the viral genome. The current Trinidadian isolates, which we got between 2007 and 2011, all fell in group C, except for 1 isolate, which fell in the Bunyama serogroup, right? Going back to the phylogeny, I wanted to point out a few interesting isolates. So if we look at group C more closely, if I zoom in on that part of the tree, you'll see, looking at the blue highlighted sequences, we have 2 interesting pairs. The first pair is at the top of the branch, having 2 isolates, TRI-712, which is a Paraparuvirus, and TRI-5972, which is an Oriboka virus. And those are 2 viruses that belong to 2 different serocomplexes within group C. So group C, because of how big it is, has different serocomplexes within it. And those two isolates belong to two different serocomplexes, but they group together with 100% identity, right? Another group of isolates I wanted to point out are at the bottom of that tree. If you look closely, there are two isolates there, TRI7121, which is a virus called Caraparu-like virus, because the serology is similar to Caraparu-virus. And below it is another isolate called TRVL34053, which is another Caraparu-like virus. Now, this virus is unassigned. It doesn't belong to any serocomplex, but in terms of phylogeny, it falls into group C, and it has serology closest to those group C viruses. One thing I want you to note is the name of those sequences. The TRI stands for the fact that those isolates were isolated in Trinidad between 2007 and 2011, and the TRVL stands for isolates that were reference isolates. So anything with TRVL would have been sequenced back in the 50s and 60s. So here we have in the second group, a sequence from the 50s being right beside a sequence from 2007, almost 5 decades later. When we looked at the different segments, we saw the same grouping. Here is the M segment. And if we zoom in on group C, we see the same association. The Caraparu-virus and the Orthobunyavirus are together, while the two Caraparu-like viruses are also together higher up in that branch. Same thing in the S segment. When we zoom in on group C, we again see a close association between Caraparu-virus TRI712 and Orthobunyavirus 5972, and we see the two Caraparu-like viruses together in the branching. Looking at those results, the Caraparu-virus TRI712 and the Orthobunyavirus TRI5972 were isolated a year apart. They were both isolated from the same vector, which was Culex portesi, and that's a known vector of encephalitis viruses, right? And our hypothesis or deduction is that both of these viruses are originated from an Orthobunyavirus from the 1950s. And the reason we say that is because in the trees that we looked at, for both isolates, there are isolates from the 1950s, which are Orthobunyaviruses, which are basal to them. Now, when you look at a phylogenetic tree, anything that is basal points at something that is ancestral to whatever branches off from that base. The Orthobunyavirus has been shown to be found in tufted capuchins, and because we have native capuchin species in Trinidad, it's quite feasible that these two viruses originated from an Orthobunyavirus. Now, that brings up an interesting question. These two viruses are in two different serocomplexes, Caraparu and Orthobunyavirus. So why are they so similar? Why are they always grouping together? And the question is, are they the same virus species? Our answer was yes, and they're both likely Orthobunyavirus, right? Now, what you'll find is that misclassification of orthobunyaviruses is not unheard of, happening due to serology. So because these complexes are so close and they have high cross-reactivity, it's quite feasible that this Caraparu-virus was misclassified and is actually an Orthobunyavirus. Another example of that was with Jonestown virus, which causes encephalitis. A study by Kinsella et al. In 2020 found that most likely a lot of those viruses were being mislabeled with La Crosse virus, right? So depending on the serology and the results you get, you can misidentify these viruses. And that is why it's very important for us to do this sort of sequencing. So through sequencing, we've shown that this isolate, which by serology would be called Caraparu, is actually an Orthobunyavirus. If we go back to the second pair of isolates I showed you in the tree, the TRLV 34053, that was isolated in 1960, and the other virus, 7121, was isolated in 2008. So that is a 48-year gap between these two Caraparu-like viruses. What we saw in the tree, and when we examined their segments, was that there was almost no microevolution across them, right? So the question then is, one, is there some kind of contamination or mislabeling between these two isolates, or are we just looking at a high level of conservation? When we looked at the different genome segments, we saw more than 99% amino acid similarity. So 99.5 in the L segment, 99.1 in the M, and 100% in the small segment. Using these, we calculated the evolutionary rate for the group C viruses as a whole using the S segment, and we got a figure of approximately 1.6 times 10 to the minus 4 substitutions per site per year. So looking at the gap between these two viruses of 48 years, if we apply that time period, we would expect at least 5 substitutions along the genome, right? Using that, if you had 5 substitutions or 5.3 between two isolates, you would expect a similarity of 99.2%. So you'd expect a divergence of 0.8. So the fact that we saw no divergence in that segment, given the mutation rate, points to a high conservation. We wouldn't say they were the same viruses simply because the other two segments did have some measurable diversity, which indicates they're not the same exact isolates, right? So a 48-year gap, but very little diversity. A third isolate I just want to mention, it was not highlighted on the tree, but it was interesting because of what we saw, is a virus called Restan virus, RESV, and this was sequenced in the 50s, and that is the code for TRLV 51144, and it belongs to group C again in what's called the Manitoba complex, right? When we look at the genome segments, those two Caraparu and Orthobunyaviruses we looked at before, it associates with them in the L and S segment. So in the L and S segment, this virus is actually outside of its complex. It's associated with these two viruses, which are actually in the other Orthobunyavirus complex, right? Which is interesting. However, when we looked at the M segment, then it turns out where we expect it to. So Restan here is seen with Nepoyo virus and Morotusu virus, which are in the Nepoyo complex, and that's part of the Manitoba complex also. That's where we expected it to be. So the question is, why is it looking like that in the LNS segments, but not in the M? And as we can see here, if we look closely at this M segment, the Nepoyo virus is basal to the branch with that Restan virus, right? So those should have been, that's the Manitoba complex, that's what we expected, but we didn't see that in the L and S. So in discussing this third isolate, what we thought was, okay, we have 3 viruses, 2 that we've shown are actually Orthobunyavirus, the 712 and the 5972, and we have this Restan virus that is associating with them with LNS. All 4 viruses, including the basal 1954 isolate from Brazil, were got from Culex portesi vectors. So they all have a common vector. The M segment is closer to a virus that was isolated in the same time, Nepuyovirus. What we think is that this reston virus, which is 1963 isolate, is an example of a natural reassortant. What would have happened would be that you would have had two contemporary species, one that's an Orthobunyavirus and the other Nepuyovirus, that ended up in the same Culex portesi, and then you had reassortment between segments where you ended up with reston virus that had L and S segments from the Orthobunya group, but an M segment from the Nepuyovirus, right? So just to summarize, the phylogenetics matches the serology. What we saw that was our 8 Trinidad isolates only fell in 5 serogroups. And out of our recent isolates that were identified in the mid-2000s, those only fell into 2 serogroups, the group C and the Bunyamura, right? So we had evidence that orthobunyaviruses species were still in circulation locally, and most of them fell into that important group C serogroup, right? The only exception was a Guiana which is not known to be pathogenic, right? And some of our isolates showed evidence of virus maintenance. So even though we had sequences that were isolated decades apart, the similarity was very similar, indicating that there was not much microevolution over that period. We also had evidence that we had possible reassortants in these group C viruses, which is always important in terms of genetic diversity and understanding how viruses can evolve. And then, you know, if they become pathogenic, you have an idea of what's happening in terms of their genome, right? Our isolates show the most variation in the M segment, and that is also the segment we saw that seemed to have been reassorted in that reston virus. And the highest similarity was across the L and S segments, right? Now, this is something that has been noted in prior research in Peribunyaviridae as a whole, not just orthobunyaviruses. And it seems to be that possibly the L and S segments may have a genetic link. One is making the polymerase, one is the nucleocapsid. And for that reason, they seem to stick together more during reassortments than the M segment, which is the one that seems to go between viruses, right? So I think we're near the end of the talk, just to acknowledge work done by the Carrington lab, particularly Alberto Geese, in sequencing these viruses, the Tesh and Weaver labs at UTMB for providing the archive samples we had and the sequencing. And just to note that this work is published in Virus Genes, right? So if anybody wants to have a read of it, they can go ahead and look at that paper.